Hello students, welcome to Botany class. Now we are seeing the replication process in the DNA. So what is meant by replication and how this replication occurs in the DNA molecules, that is in the DNA strands. Okay, so first of all, we have to see where this replication will be done and how it will be happening and what could be the sources, otherwise the enzymes and the other requirements for doing this replication process. So the replication process is the proof of the semi-conservative nature of DNA. So what the Watson and Crick, especially Crick, said that DNA is a semi-conservative in nature. Semi-conservative in nature means it will be opening only half of the helix and the rest of the half will be like that, and it starts replicating, making its duplicate copy. That's the Crick said. So here we are seeing the same one with the experimental proof. Okay, so here what we are saying, DNA can do the replication. That means it can make its original copy. Okay, so how this replication occurs and what are the things behind that is necessary for the organism. For doing the replication, it needs the one enzyme, that is the DNA polymerase enzyme. DNA-dependent DNA polymerase enzyme is necessary for this process. After that, it needs the primers. And after that, that is the RNA primer is needed for this. Okay. And after that, it needs the ligase enzyme. Ligase enzyme is needed for this. So like this, it needs these three things for doing this replication process. These are the mandatory steps. So where the replication occurs and is this necessary for the DNA and how the genetic information is carrying out as it is without any change to the next generation. That is with the replication process only what we can say. So in this replication process, the first of all, this DNA-dependent DNA polymerase enzyme, that is the DNA polymerase enzyme, will search the site in the DNA. That is the ORI site, origin of replication site. ORI site means origin of replication site. And this will be present in on the DNA strands. So this DNA polymerase enzyme searches this site on the DNA strand first. Then that will be identified and that part will be occupied with the DNA polymerase enzyme and the RNA primers. And they release some enzymes which will be with they are combining with the DNA polymerase enzyme and occupies that site. Immediately, the rest of the DNA strand will unwind and makes itself as a opened type of the DNA strand. Like this, the DNA strand will be detached from one another. That is this is the ORI site. Okay. Now what happens? Once the DNA polymerase enzyme is attaching at the otherwise sticking at the ORI site, immediately the DNA polymerase enzyme only making the RNA primers, mini primers will be there, so that it makes the another strand of the DNA, and these RNA primers are gets separated from the anchor, that is the where this DNA has to be doing this replication process. Now in this DNA strands, it is having the ends, that is the 5' to 3' and the 3' to 5'. So here the one strand is this is going from the 3 prime to 5 prime, and this is coming from the 3 prime to 5 prime direction like this. Okay, so usually in the DNA, one strand is called as the template and another strand is called as the complementary. But during the replication process, both the strands are called as the template strands because there is no complementary strand to one another. Both will act as a template strands. Once this template strand was made, immediately the DNA polymerase enzyme, along with the help of the RNA primers, it starts making the new empty DNA strand. And this strand has to be formed from the opened end of the strand of 3 prime to 5 prime end. Okay, so this strand is, now we have to take the ends of the strands from the open end, not from this one, where the unwinded DNA strand is an opened DNA strand. This here only we have to take. Why here only? Because here about this is the DNA polymerase enzyme is sticking to this ORI site. So we have to calculate this site as the first one what we have to read. So once the DNA strand is having the 3 prime to 5 prime strand, that is the template strand, in that strand only, first of all, a new DNA strand was made from the 5 prime to 3 prime direction. That is the continuous manner. And this starts with the RNA primer. After that, DNA polymerase enzyme starts making the elongation of the DNA strand. And on this, the codes were written by the template strand. What are the codes are where, whatever are present, the complementary codes will be written on this 5 prime to 3 prime strand of the DNA. So this strand is called as the leading strand. This is called as the leading strand. And on the another hand, the another strand which is going from the open end is the 5 prime. 5 prime to 3 prime direction. This strand will be producing a new DNA strand in the 5 prime to 3 prime direction. That means from here to this side, open end. So this is the discontinuous strand is formed. This is called as the lagging strand. The discontinuous strand is called as lagging strand. Like this, the leading and the lagging strands, that continuous and the discontinuous strands are formed. And these discontinuous strands are called as the, this was the first invented by the Okazaki. So we will call these fragments as the Okazaki fragments. Okazaki fragments. This one they will ask in the match the following in both EAMCET and in the NEET exam, Okazaki fragments. So they will ask in the one row as the Okazaki fragments, and here they are formed in a discontinuous manner during the replication process. That is the constant scenario for the Okazaki fragments. And these Okazaki fragments, whatever may be the fragment, which has to be formed from the DNA strand, need to be formed from the 5 prime to 3 prime direction. And for forming this strands, what I said, RNA primers are needed. And much more energy is also required for this process, as it needs to make a new strand which where the codes has to be written. So the DNA here the DNA will give you the supplies much more energy for this, and one RNA primer is needed for making the 5 prime to 3 prime, that is the leading strand. And for the lagging strand, that is formed on the template strand moving from the 5 prime to 3 prime direction, where the Okazaki fragment, that is the lagging strand, was formed in the 5 prime to 3 prime direction. And this one is needed with the many RNA primers, because each and every fragment need to be made separately. This is 5 prime to 3 prime, and this is 5 prime to 3 prime, this is 5 prime to 3 prime, this is 5 prime to 3 prime. Like that, many fragments are formed. So many RNA molecules, RNA primers are needed over here. So that's the difference between the leading and the lagging strand. Like this, the strands were made after some time. Again, this gap was filled again. And that means with these two strands were joined, Okazaki fragments, to make a continuous strand. If any code was missing, then an empty strand was made. On that, the code was written, and this was joined with the ligase enzyme, the DNA fragments, Okazaki fragments. That was done only with the checking and making the new nucleotides, that is the new codes on the lagging strand, as well as making an empty strand, and the joining of the enzyme, that is the ligase, is going to be produced. Everything was done by the DNA polymerase enzyme. This is having that much of great capacity to produce the enzyme, whatever it is needed for that purpose of the situation in the replication process. Okay, this is the replication. So it looks like a fork-like occurrence. So it is called as the replication fork. Once it was finished, then it divides and makes this will go into the new offspring, one offspring, and this will go into the another offspring. So both the offsprings are having the same parental genotype and the same behavior and the same physical appearance. That is like that. The genetic characters are going from the parents to the offsprings by the replication process. Okay, and we will see the next topic in the next video class, right? Thank you.